Can you all? Okay. So it's 6 o'clock. We will get started right on time. So if you can find a seat, get settled, get comfortable. We're in for a wonderful, wonderful talk tonight. A few more people coming in. There's a lot of seats right up the front. You don't want to have to go through the rows there. So come down this way. Right.
How about that?
Located. I have the privilege of welcoming you all to the first seminar of the spring semester. My name is Adeliza Flores. I teach chemistry here, and I'm part of the planning team as well. Before we get started with our speakers here, I'm going to try to do a little bit of a brief history of the seminar series and a short list of acknowledgement. I'm going to try to keep it short. But first of all, this is our 27th year, I believe. Sorry, 7th year. This is our this is our 7th year, our 27th seminar. Every year we have 4 seminars, and a brief history. The catalyst for the seminar, and as far as I know, she's not all used up, is Nan Ho. She was a former biology faculty. She's still a biologist, very much. But she's now our dean for the Math, Science, Engineering, Public Safety Division. And she did some work at the lab, and she really enjoyed all the talks over there. She wanted to bring them over here because, of course, it's not very easy for us, students in particular, to go there and participate in these seminars. So that's that. And let's see, so the title of the seminar series encapsulates what it's trying to do and to bring to the campus. So the long title is Lawrence Livermore National Lab LPC Seminar Science and Engineering Seminar Series, How Science is Done from Theory to Practice. Anyway, long title. So before we start, I want to thank some people in the planning team. From the Lawrence Livermore National Lab, of course, we have Dr. Chris Culp. I don't think she's here tonight, but she's a group leader in the Biosciences and Biotechnology Division. And there's also Joanna Albalado, if she's here. She's way in the back there. She's the Education Outreach Manager. She's been helping bring these great speakers to our campus. Let's see. From the LPC side, we have Nan Ho, of course, who was the original brains of the seminar series, helping the planning team. Another team member is Kelsey Watt, who is our STEM coordinator, so she's right over there. Let's see, we have David Lee, who joined our team this year. He's doing all the AV stuff, so we're really happy about that. So he's right back there. And then the rest of the IT group as well. Now we have some new planning team members, and I know at least 2 of them are here. There's Mike Ansel from chemistry. A lot of you probably know him. He's right back there. And Dr. Robin Rehagen, all the back there. She's in physics, so she's joined our team. And then there's Mr. Kislow. Some of you may know him. He's engineering. I don't think I see him here. Anyway, they're new members of our team. Let's see. I have to look at my list. I do want to acknowledge the faculty who are here, who brought their students and the staff. Thank you all for coming, and of course the students. And lastly, I wanted to thank Dr. Neil Eli is here. So once in a while he pops up. He's our former dean. He was an original member of the team. And then Dr. Lisa Everett also was a former dean. And then last, from the lab, part of the original team, Dr. Beth Vitalis and Dick Farnsworth. Now on to our speakers today. Our Nano-Bot team consists of Dr. Matt Coleman and Dr. Amy Rasley. They're sitting over here. Let me say a few things about them before I let them take the stage here. Dr. Coleman is a senior biomedical staff scientist at the lab, Lawrence Livermore National Lab, but he's also an adjunct professor in the Department of Radiation Oncology at UC Davis in the School of Medicine. His PhD is in molecular biochemistry and cellular biology from Boston University. He has authored lots and lots of publications, but the main focus of his work is the development of advanced biochemical techniques using nanoparticles. Dr. Rasley, she's a senior staff scientist at the Host Pathogen Biology Group within the Biosciences and Biotechnology Division at the lab. Her bachelor's is in zoology from Weber State University in Utah, and her PhD is in immunology from the University of North Carolina in Charlotte. She has studied immune responses within the central nervous system during bacterial and viral infections, but currently she is working on the use of nanoparticles and its applications in immune response systems such as vaccines. Today they will be talking about how they've used synthetic and molecular biology tools to create small nanomachines, which they call Nano-Bots. And these have potential practical applications to vaccines, drug delivery, and energy needs. So without further ado, let's give them a big hand. Dr. Coleman.
Can everybody hear me? All right, if I need a mic, I'll put a mic. So I'm Matt Coleman. I'm from Lawrence, Livermore, and today we're going to talk about our nanobots. And this is part of a talk that developed out of the science on Saturday called Ingenious Inventions. And we're going to talk a little bit about these pretty pictures, and I'm going to take you through an introduction, pretty much get us all on the same page with some of the tools, how do we visualize, make these things. And then my colleague Amy's going to stand up here and tell you what we can actually do with these things and give you real-life examples. So the outline for today is, so what are nanobots? Visualization of nanobots, how can we actually see these things and manipulate them? And then what are the things that we can do with these nanobots? All right, so for the first thing I want to do is define this term, robot. And from the Oxford English Dictionary, the term robot basically means a machine that's capable of doing something over and over, very repetitively, and you can trust it to do that again and again and again. And when we think about robots, if you're old like me, right, I think of robots, I get Lost in Space. Anybody remember Lost in Space? All right. Everybody with gray hair has their hand up. And I mean, if you think about Star Wars, yeah, there's lots of robots and everything, and they have different functions and different capabilities. But in biology, we want to think a little bit like robots, but we've got to think of it in a different way. And how can we make biological material that are going to be able to do things over and over and over again? And so first of all, also we got to talk about at what scale. You know, do we want to work at a meter scale or a very small nanometer scale? And what is that? What are the biological materials that we're going to use to do exactly this engineering with the biology? And then what can we do? What questions can we address with this material? So first of all, I just want to stop and take a moment to talk about scale. So everybody knows, you know, what a meter is. Basically is 3 feet. But we want to work at a much smaller scale. And so we can start by stepping down the scale and just remind ourselves then what is really small. So a meter, or 1/1000th of a meter, is a millimeter, and ants are about 5 millimeters. We can step down a little bit more and look at an insect eye, such as a fly eye, that's around 1 millimeter. We can go down to step down another scale then to micrometers. So micrometers, 1/1,000,000th of a meter. And we can think of human hair, or maybe some collection of red blood cells. But we need to go even smaller than that. So we want to get down to 1/1000,000th or a nanometer scale. And we want to be working with actual sort of proteins, which are bigger than atoms, which are collections of atoms, but are tenfold or more in this range. And so what we're going to talk about today is in this nanometer scale, and it's going to be, this little cartoon is sort of depicting this 10 to 25 nanometer nanolipoprotein particle. And so I want to talk a little bit about what is this nanolipoprotein particle. Well, this cartoon scale here is showing you and meant to represent that nanolipoparticle, where we have a belt, and that belt is going to be made up of individual proteins, and then surrounded and within that belt, what we're going to have are phospholipids or fats and detergents, and as we work through this, we'll put other things in there as well. And this whole thing is going to have a height of about 5 or 6 nanometers, and it makes up this little disc. So this cartoon is what I'm showing you here. We can get a little bit more detail. And now what you're looking at, though, is that that belt is actually made up of proteins that form alpha helical structure. And there's 2 proteins here. So there's protein 1 and 2. They're the same protein. And they're just sort of wrapped around those lipids. So in white are the different lipids. There's about 160 to 250 lipid molecules, about 2 to 4 proteins per disc. This can really depend on the type of disc we make and the proteins we use. And it has a relationship actually to what's going on in your human body, because these are actually very common proteins that we're working with. And people have known about these for over 40 years. So if you ever gone to the doctor and they said to you, we need to look at what your HDL level is and your LDL level. Has anybody heard that before from the doctor? And they're asking specifically to measure a component that is part of those particles, and that's your cholesterol. Well, there's a protein that comes together to form these HDL, or high-density lipid particles, or low-density lipid particles, and it's called an ApoA protein. And that model I showed you, so I have one in my hand. I'm not sure everybody's going to be able to see it. But you see I have these little, what can actually be like sort of bracelets here. So that's those 2 proteins. They come together, and it's a nice little 3D printed model that you're welcome to come up and play with. And here's my lipids. So these proteins kind of come along, wrap around, and form this little disc-like structure. And now this disc-like structure has grabbed lipids, but now it can also grab cholesterol. And if it's ApoA1, it then becomes part of that HDL complex that picks up and transports cholesterol and lipids. If it's an ApoE variant, then it's going to be part of those HDL particles. But this little disc is a transition state to forming those particles for transport in your body. And my next example actually shows you that what this little particle can do. So what I'm showing you are, there we go. So what I'm showing you are 2 tubes. This is a tube with just lipids or fats in it. And what we basically do is we take those lipids and fats, we add the nanolipid particle, and hopefully you can all see it, that it's now clear. This particle is actually so capable of going in and grabbing and picking up and absorbing all of that material. And when you look at my model, you'll actually notice that it's not a solid ring. There's a break in this ring. This ring actually has flexibility to open up. So it sort of can change its size depending on how much it needs to actually transport. All right, so that's our basic building block, what we call the scaffold that we're going to use to develop our nanobots. So the next few slides I want to talk about, though, are what are the techniques we actually use in the lab so we can see these things. And one of the first and most common techniques is gel electrophoresis. And in this case, polyacrylamide gel electrophoresis. And what you're looking at here is a polyacrylamide or gel-like matrix. It's kind of like Jell-O, really, in material that's clear, where we can load proteins on the top, and we can use an occurrence. So up here is a negative pole, positive pole. And when we treat the proteins, we can run them through this matrix and separate them based on size. So they'll run through here under a current, and each band will represent a different protein of a different size. And in our case, when we look at the apolipoprotein and we stain this matrix, here is a molecular weight marker where this is 200,000 kilodaltons, is the size of the protein down to about 6. It's very light, but there's a protein here. And this is what the apolipoprotein looks like when we purify it on a jet. So this is one way in which we can have a lot of the protein and we can actually see it on the gel. But we also have to have ways that we can purify it and sort it, not as an individual protein like I just showed you on a gel, but as it makes a complex. And that next technique that we use is something that's called size exclusion chromatography. So basically what we do is we take a column. Once again, we're going to fill it with another type of gel matrix. And that gel matrix allows small proteins to pass in through it, through a pore, but it excludes big proteins. So hence the name exclusion chromatography. And what happens is as material moves through, the larger proteins are excluded from the pore, so they actually come through much faster. The small proteins have to go into the pores, come out of the pores, go to the next bead, and do the same thing over and over, so they elute much slower. And this is shown over here in a little diagram or chromatograph of the large protein complex coming off versus the individual protein complex coming out much later over time. So large things come out fast, small things come out slow. And here's an example of that for making a nanolipoprotein particle. So once again, this is a measure of the amount of protein you have over time of collecting those fractions. And so all the large things, actually in this case, these are free lipids, or what are called liposomes. They tend to be around 80 to 150 nanometers in size. Then our NLPs come out. They tend to be that 10 to 25 nanometers in size. And then any free protein that didn't absorb any of the lipid or it did not form into the NLPs comes out somewhere much later. So these are your much smaller globular proteins. And then now I can put that together with a gel. And here we can see a single band on what's called a native gel that shows us that protein is formed into a complex. Okay, the other technique that we're going to have some data and show you some data on is atomic force microscopy. So atomic force microscopy is a biophysical technique where basically we have a little probe. If you imagine that's my finger, and there's a particle here, and I'm going to raster across the surface. And as I come close to something, I'm going to actually push up on it and force field over it and notice that it's there. So I'm going to look at the topology on a surface of what these particles look like. And this first diagram is just kind of displaying that for you, where the tip rasters across the surface. I hit an NLP, I bump up a little, then I bump back down, and then I get a little graph of what happened. And actual data in an NLP and B shown here. Now in blue are where the NLPs are actually sitting down on that surface, and below that I get a little diagram of everything I see. And you'll see that I get things that are 20 nanometers across roughly, and 5 nanometers in height. And the important thing is what I'm really measuring here is the topology, or that change in height. When I get over to an NLP here in picture C, why these are not blue versus this picture here is that these NLPs look like little mountains because they actually have things in them. And we're going to talk about that a little later in the talk. And they look like little mountains, so the topology is actually shifted from 5 nanometers to 5 nanometers plus as we modify these nanoparticles. And then the last technique I'm going to talk a little bit about that allows us to actually look in and see these particles is electron microscopy. So electron microscopy works over a range of 100 microns down to a tenth of a nanometer. And it's very much like light microscopy, except for we're going to use electrons rather than photons to see things. And in our case, we're usually using this to image down around 5 angstroms. So in 8 here, what I'm showing you is these nanolittle protein particles, and they do something called cookie stacking. So if you can see this little light image here, they're all stacked up just like Oreo cookies. And there's one all alone over by itself. Here's another image of a different type of nanolipoprotein particle. And this is actually the nanolipoprotein particle sitting next to a virus particle. These railroad track-like or road-like structures are a virus. And then this is just a close-up. And you can see it has some blob and density to it. But you can see that from these images, it's a disc-like shape. Right? And we get an approximation of size. All right, so how do we make these particles? And so we know how to visualize them, but how do we actually make them? Well, we're not going to talk about this today, but we actually do make 3 different types of particles. One is this nanolipoprotein particle. We have a telodendromer disc that's a totally synthetic process. And then we have a hybrid process where we can combine these 2 things to make a telo-NLP disc. And today we're going to really focus on how we make these, the NLPs, for the bodies of our nanobots. So the first method is called the traditional assembly. We take purified protein, we mix that with lipids and a detergent or surfactant, such as Coleman, and then we dialyze away the Coleman. And the Coleman allows the protein and the lipids to come together to interact and form our little nanolipoprotein particles. And this whole process takes anywhere from, if you have the proteins in the fridge and the lipids, and you can just mix it all up, this can be done in a couple of days. If we do a synthetic nanoparticle, we can actually do that in a matter of minutes, 15 minutes. And that's just by mixing our chemistry with our lipids to get this product. And then finally, we have something that's called a cell-free approach that we're going to talk a lot more about. And like that traditional assembly, we have to mix in lipids, but we're going to mix in plasmids or DNA and nucleic acids to make this particle. And very much like that synthetic approach that I just talked about, this can be done in minutes to hours, rapidly increasing the time in which you can make one of these nanoparticles. All right. So I'm going to focus a little more on what is this cell-free method. So, and I just want to remind you that what we're taught in biology and what's the dogma of biology is DNA makes RNA makes protein, and that process is something called transcription, where DNA makes the RNA from the DNA template using enzymes to accomplish that, and then that RNA is passed off to ribosome to read that RNA, coordinate it with the tRNA, and make a protein. And together, that's called coupled transcription and translation, and that can all be a single test tube for some of the data I'm going to show you. So that whole process of cell-free and the test tube is we take the ribosomes from a microorganism, E. coli, and we have this cell lysate mixture that has our ribosomes and our tRNAs, our amino acids and everything. We add a DNA template, put it in a test tube, out comes a protein, and that same process can be done with the NLP. So we take our cell lysate, but now we need to add lipids, and we add DNA that encodes this belt protein, and wait some time, and out comes formed NLP, and this can be done in basically 1 hour, in full from production to purification all in 1 day. So how are we going to use that scaffold then to make our nanobots? Well, we can chemically add moieties to either the lipids or the belt protein, and those can be attached through chemical modification, or we can actually produce receptors that embed themselves into this membrane or sit on top of this membrane, and that becomes our sort of nanobot in each case. So if this is a functional entity here that does something, then this scaffold can then support that and carry that into an animal or into a body. Or we can make also hybrids of that where we can add chemical entities, receptor proteins, and other surface attached proteins to give us a function. All right? So once we add all those functions then, that's what we're calling our sort of nanobot. We take our NLP, we actually modify it by either adding other functional proteins or chemical entities to it so that we can do biochemistry, make sensors, develop vaccines, do drug delivery, or cancer imaging. And today what we're really going to focus on are some examples in biochemistry, sensors, and vaccines.
Well, thanks everybody for being here. I'll talk a little bit about how we use these particles. As Matt mentioned, we're going to go through 3 examples. So we're going to talk about how we use these particles for biochemistry, how we use them for sensors, and then finally how we're using them for vaccines. Can everybody hear me?
No.
Okay, I think, is that better? Okay. Okay, so the first example is a basic biochemistry example. If any of you have flown into or out of San Francisco International and you've had the luck to have a window seat, you may have noticed these beautiful ponds that are kind of throughout the South Bay. So these are actually the Cargill salt ponds. And these colors originate from the presence of microorganisms. In particular, a microorganism known as Halobacterium salinarum. It's actually not a bacteria, it's a member of the archaea. But specifically, the color comes from the presence of a protein that resides in the membrane of these organisms called bacteriorhodopsin. It's a pigmented protein, so that's why you see this beautiful red color. Incidentally, you can actually see these salt ponds from space, which I thought was pretty cool. So this protein, bacteriorhodopsin, comprises a large chunk of the membrane of this organism. And it essentially functions as a proton pump, allowing the organism to convert light to energy. And so this protein is a pretty, I would say, a representative membrane protein. So it's been very well studied. Membrane proteins in general are very difficult to work with, or can be difficult to work with. If you try to use more traditional approaches to express membrane proteins, such as using an E. coli expression system, you can run into problems with solubility. Sometimes the proteins misfold, sometimes they aggregate. So they can be challenging to study. So how do we study bacteriorhodopsin and membrane proteins in general using our nanoparticles and making these nanobots? So again, as Matt mentioned, we do this synthetically in a test tube. In this case, we have our cell lysase. It's providing our machinery that we need to make these proteins. We have our lipids, and then we have 2 DNA templates in there. One that encodes the scaffold protein, which makes the bell, and the other that's going to encode the bacteriorhodopsin DNA. And so what happens in the end is that you get the formation of this nanobot. So you have the nanoparticle, and embedded within that particle, you have your membrane protein. So this process doesn't actually happen, within minutes. In the lab, when you make these types of particles, it can take up to 5 hours. And it's shown up here. And you can see as time progresses, you see that you get this characteristic color showing in your tube, which is the bacteriorhodopsin. So you know that it's been expressed and it's inserted into your NLP, so you have a functional nanobot here. This is a great protein to study because of this color change, so it makes it very convenient. But in general, the NLPs have been used for a variety of membrane proteins to promote their solubility so that they can be studied. And this is a great example of that. So what I'm showing you here are 2 tubes. This tube just has bacteriorhodopsin protein, so there are no NLPs in here, no nanoparticles. This tube has the bacteriorhodopsin with the nanoparticles, so this is the nanobot in this tube. And you can see that the pink color is distributed throughout that tube. So this protein is soluble, it's in solution, whereas this protein without the NLP, so no nanobot, is not soluble. It's precipitated out of solution. You can actually visualize these, as Matt mentioned, using a variety of techniques. In this case, what I'm showing you is an electron micrograph. And you can see the disc, the NLP here, with the bacteriorhodopsin membrane protein embedded within that. And this is just showing you a computer model using this data. So this is using these NLPs for studying membrane proteins is a very powerful tool, because you're essentially providing that membrane protein a native environment that it likes, which is a lipid membrane. And we can provide that synthetically using our NLPs. So as I mentioned, this is not just specific for bacteriorhodopsin. The presence of NLPs and having your membrane protein embedded within that NLP enhances the solubility of a number of different membrane proteins with various transmembrane domains. So what I'm showing you here are just membrane proteins with various transmembrane domains. So 2, 3, 4, 7, that's also 7. This is bacteriorhodopsin here. And what you're looking at is the presence of the membrane protein alone, this first bar, the membrane protein with lipid. So you've essentially just added lipid and added membrane protein. Or you have essentially a nanobot. So your membrane protein embedded into your NLP, and that's this bar here. So this is percent solubility. And you can see that when you have these nanobots, you're increasing the solubility of these membrane proteins pretty dramatically in most cases. So again, very powerful tool to use, can be made in a test tube. So what are some of the other things that we can do? I'm going to talk to you about how we label these particles. So in this case, this is Wade. He's not here, I don't think. Wade, are you here? No. Okay. So Wade is a postdoc who makes these particles, and he's demonstrating how he makes a fluorescently labeled NLP. So in this case, again, you have your machinery, your template DNA goes into the tube. And in this case, May has made green fluorescent protein, and that is visible here in this tube. So this is our labeled NLP here, and this tube is not labeled. So this is the control, essentially. So green fluorescent protein is used a lot in biology. It has a long and illustrious history. So the folks that described GFP and worked with GFP received the Nobel Prize in Chemistry for that. So it's something we use all the time. It's a very standard tool, but we don't want to forget that it actually has a cool history. So Wade has made this fluorescent particle. How do we use these types of nanobots? All right, so this is an example of how we use these labeled nanobots for sensing. So in this case, let's say that we want to sense a specific microorganism, for example. So we make our nanobot where we have a cell lysate mixture, our lipids, we have our scaffold DNA, gives us the band, and then we have our sensor DNA. So this encodes a protein that has a binding partner that this particular microorganism secretes. Okay? And we can actually verify that we have made our nanobot sensor using this gel electrophoresis technique that Matt has described to you, where we can essentially detect the presence of two bands, this band indicating our nanosensor, our protein, and this band here indicating the presence of the scaffold. So this is our intact and functional sensor. And you can also use atomic force microscopy, as Matt described, to ensure that you actually have your protein embedded in your nanoparticle. And again, what you're looking at essentially is a change in height. So you're ratcheting over the surface of these of the nanoparticles as they are laid on the surface, and you're looking for change in height, which you can see. So empty NLPs are here in blue. Okay, so they're about 5 nanometers in height. And as you incorporate these membrane proteins into them, you get a shift in height, so they become bigger. And that's shown here. So it's just another way to verify that, yes, this protein is in fact in your nanobot. So how do we actually know if our Nano-Bot sensor is doing what we want it to do? So essentially we're looking for a binding event. Again, we have a protein in here. This is our sensor protein. It's going to bind to a protein that this microorganism is secreting. If that binding event occurs, you're going to see something like this, where as you increase the concentration of your Nano-Bot sensor, you're going to get more and more binding events that occur until you max out and reach 100%. If our nanosensor is not binding its partner, you won't see this. You'll see pretty much a flat line here, even if you increase the concentration. So in this case, we have our sensor. We have our labeled, GFP-labeled protein in our sensor, and we're going to try to detect a cognate binding partner secreted by this microorganism here. What does that actually look like? So this is real data, and essentially it looks very similar to the curve that I showed you. Again, it's a concentration-dependent event, so as you increase the concentration of your Nano-Bot sensor, you get more binding. And the actual data points are shown in stars here. So at some point, as you increase the concentration, you will max out essentially, and you will saturate all of your binding sites, so the curve will go flat. And this line through is just the best fit to that data. So that's what actual data looks like that comes out of these types of studies. Okay, so I'll talk a little bit about how we use this for vaccines. And why are vaccines important? So vaccines really are one of the hallmarks of modern medicine. They work very well. We know why they work for the most part. And just to give you an example, this is measles. So we have measles cases shown here, and this is time or year down here. And what you see is when the measles vaccine was introduced in 1968, the number of cases of measles dropped dramatically to almost zero. And that correlates with the number of deaths as well. So vaccines are very powerful. Of course, they can always be improved. So our questions were, can we use nanobots to essentially make them look like a microbe? We can add things to them. We know that we can impart functionality. So the question is, can we make them look like a microbe? And is that good enough to stimulate an immune response? Does that, and furthermore, does that immune response provide protection from actual disease? And is it safe and well tolerated? So vaccines can have side effects, as everybody knows. So this would be a big bonus if we could develop vaccines that we could do it faster and make them safer. So one of the first questions that we wanted to know, if we're going to put these types of particles into an animal, we wanted to know where they go, what happens to these particles. So in order to do that, we fluorescently labeled our NLP. So we have our fluorescent nanobot here. We injected mice. In this case, we injected mice in the stomach, or what we call intraperitoneal. And then we imaged them using a fluorescent imager. We just asked the question, where do we see this fluorescent signal in the animal? So this is what the data actually looks like. This is a mouse laying on its stomach. So this fluorescent signal is coming from the nanobot. You can see the intensity of that signal is greatest where you see white and yellow. And it's less intense as you go from this orange to black. So this is looking at the stomach. You can see this very intense signal here. This is the bladder. So definitely these particles are being filtered through the kidney. The animal will ultimately just pee that out. If you lay the animal on the side, you have a nice view of this right here, which is the kidney. And again, very intense signal there. And this is just the back side, so you see both kidneys here. Kidneys are retroperitoneal, so that's a great way to look at the kidney. So when you inject these particles intraperitoneal or into the stomach, for the most part, they're going to be secreted through the kidney and also through the liver. So if you take this animal and you cut it open and take the organs out and image the individual organs, then you can see again, very, very high signal in the kidney, pretty high signal in the liver, a little bit in the spleen, and not very much in the lung at all. So this is mainly going to be, again, secreted by the kidneys. what if you administer them a different way? Do you see this a similar profile? So another way that we typically administer these particles is intranasally. So essentially you take a pipette and you put the liquid on the nose of the mouse. Mice are natural nose breathers, so they will just breathe that liquid in. So in this case, we gave the animal fluorescently labeled Nano-Bots in the nose, and we did similar types of imaging experiments. So what I'm showing you is this profile intranasally, and I'm comparing it with this intraperitoneal or in the stomach. So you can see that it's a different profile depending on how you administer these particles. So you don't see a very high signal in the kidney when you give these intranasally, but you see a really high signal in the lung, not too surprising. But that signal is retained over a course of days. So the take home is that the way that you administer these particles very much impacts how they're distributed in the body. What I'm showing you here are just histograms that are quantifying this fluorescence intensity from our imaging studies. So in this case, in B, we've given these fluorescent Nano-Bots intravenously. So you basically inject these directly into the tail vein of the animal. And you can see again, very high signal in kidney, a little bit in liver, not much in spleen or lung. And that's very similar to what we saw intraperitoneal, which is shown here. There's a little bit more in spleen, liver, and lung. If you give it intramuscularly, so just right into the muscle, we usually do it in the hind limb of the animal. Again, pretty similar profile. Most of the fluorescent signal is found in the kidney. Similar for subcutaneous, so just underneath the skin. And again, there's our intranasal profile, which looks very different from the rest of these, where you see a very high signal in the lung, and that signal is retained over time. So this is important to understand if you want to use these for vaccines, or if you want to use them for drug delivery, for example, you need to understand where they go. If you're trying to target something to the lung, then you probably don't want to give it intravenously, for example. Okay, so one of the other big questions is, are these particles toxic? So this is actually a big one for anything that you want to ultimately put into people. You have to demonstrate that toxicity is not an issue. And this has been an issue in general for nanoparticles. And there are various ways that people mitigate that toxicity, but it has been an inherent problem in the field. So in this case, we essentially adopted an acute toxicity protocol developed at National Institutes of Health. And this protocol was essentially to take these Nano-Bots and inject mice daily for 14 days. So every day for 14 days, they got injected with these Nano-Bots. And then at the end of that 14-day period, well, we weigh them daily. And then at the end of that 14-day period, we excise tissues, which we also weigh. And then we do histology to look at the microstructure of those tissues to see if there's any evidence of any kind of toxicity. So the answer is there's not. So we use both male and female animals just to make sure that we're not seeing any differences due to sex of the animal. What you're looking at here is weight of the animal over time. And again, this was a 2-week experiment. And in the males and the females, you don't see their weights really changing at all. So the animals aren't losing weight. That's one of the first signs that an animal is sick. They stop eating and they lose weight. So that's a very easy thing to measure. So in this case, weights don't change, animals were eating fine, so that's great. Now at the end of the experiment, we excised organs. In this case, we looked at liver, kidney, lung, and spleen. We looked at males and females again, and we weighed those organs just to make sure that there was no difference in weight between them. That would also tell us something about whether there was a potential immune response. So spleens tend to get bigger when you are having an immune response there. But again, no change in weight of the organs in males or females. This is PBS, so these animals essentially receive just a saline solution, that's a control, and everything pretty much looks like your control. So no real impact on the organs at that level. And then just to make sure, we did histology. So what I'm showing you here is this is actual liver that we took out. We sliced it up into very thin slices, we stained it, and then we looked at the microstructure. So this is, again, our control animal, so it didn't receive the Nano-Bot. This is the animal that received the nanobot in the stomach or an animal that received it in the nose. And again, liver looks very similar to control. So no obvious disruption of the microstructure of the organ. So we can conclude that these particles themselves are not toxic. So that's good news because we want to use them for vaccines, for example, or drug delivery. So how do we use them for vaccine development? So the first thing we have to do is identify an organism that you want to target. Okay, so it could be you want to make a new influenza vaccine, or you want to make a vaccine against whatever. And once you do that, you need to identify a target from this organism. So what's your protein target going to be? That's actually not an easy thing to do. That part actually takes a lot of time and basic research. But once you've identified your protein, you need to express it. Also, not a small thing. So you have your protein that you've expressed, and then you need one more thing. You need an immune-stimulating molecule, or what we call an adjuvant. So for the vaccines that you and I get, the adjuvant is alum. Alum has been around for decades. It's one of the few approved adjuvants that we have. It's also not great, so we don't use alum. We use other things. But you need that immune-stimulating molecule. Okay, so now you have your isolated protein, you have your immune-stimulating molecule, and you can put these all on the NLP and make this functional nanobot vaccine. What's really great is that on one individual particle, you have all of the components necessary to enhance an immune response. So, what do we do next? We take these nanobot vaccines, we vaccinate our mice, and then we ask the question over time, what happens to this animal's immune response? How is it responding to our vaccine? So these experiments typically take up to 4 months. We give more than one vaccination. So we usually do an initial, what we call a prime, and then a boost. So if you've ever had vaccines where you have to go back and get another shot, that's called a booster. That's a very typical thing to do. And that's kind of a regimen that we use pretty routinely. So how do we measure the immune response in a mouse? It's actually almost the same as how we measure it in humans, and you and I. But you vaccinate your animal. Time, you can draw blood from this animal. And you can look for specific proteins called antibodies. Those antibodies that you're looking for should be specific to that protein that was part of your nanobot vaccine. So what does that actually look like? So we can quantify the presence of those antibodies, or what we call a titer, from an animal. In this case, this animal was vaccinated and then boosted twice. We sampled blood at 4 weeks, 8 weeks, 12 weeks, and 32 weeks. So this was actually quite a long time point. The lifespan of a lab mouse is about 2 years. So 32 weeks is like a middle-aged mouse, basically. We were asking the question, first of all, what kind of a titer level do we see in this animal? And how stable is it over time? In other words, does it go up and decline? Does the titer come up and then remain constant? So that pattern is kind of important. If you can keep a titer high, you are less likely to need to boost, which is nice, because nobody wants to get more than one shot if they don't need it. So this particular animal had a really robust response. And you can compare that to its control companion mouse that just received saline, so no nanobot vaccine. You don't see really a measurable titer there. So it had a great response, and that response was sustained over time, which is very important, so indicating that the stability of that immune response is good, out to 32 weeks. So measuring titers, or levels of antibodies present in the blood, is just one way that we can assess the immune response. There are many other ways, but this is a very good way. Ultimately, the way that you want to test your vaccine is to infect that animal with the organism you're trying to protect against, and ask the question, does that animal survive? Does it get sick and succumb to infection? And these are the types of experiments that we do. So this is really your ultimate demonstration that you have an efficacious vaccine. Oftentimes what we see is you can measure the immune response, and by those measurements, the immune response looks great. You have high titers, you have a number of other things that are positive readouts, for immune response, but then you do this experiment and you don't see any protection. So then you go back and you iterate, and it's very incremental that way. So, just to summarize, NLPs can be produced in a single tube. So this whole process can be done synthetically to make these particles. They can be functionalized to do a number of different things and be used in a number of different applications. And we've just described a few of these to you. Those few examples that I've given you have been actual individual projects that have spanned 2 or 3 years. So this is really a snapshot of the work that we've done in this area. So there's a lot of potential with these nanobots in a lot of applications. So just to give you an idea of kind of the effort, this has been a long-standing effort in the lab, over 11 years probably. A number of people have worked on it and are continuing to work on it. So Matt and I are here talking to you today, but there are a number of people that work on this routinely with us. So I would just want to say thanks to all of these guys. And then please ask questions. We're happy to take them. Thank you. So the question was, we have control over the size of these particles.
Yeah, so, yes, and actually the size is determined by the ratio of the protein to the number of lipid particles. And we can precisely control that and use the same proteins to make different sized particles.
I love that though, when you inject these particles into these cells, you're likely going to have incorporation of endogenous solids, so they will change shape. You probably do get that from vivo. I would imagine that happens. It's probably a very dynamic process. So outside of a body, yes, we can make them a specific size. But once they get into an animal, I would say all bugs are out there. Yeah.
So regarding the vaccines and how you choose the secreted proteins, for those proteins
So that's actually a little bit tricky. For a lot of the organisms that we care about with a biosecurity focus, obviously we care about other things too, but that's kind of our mission at the lab. There's not a lot known about how these organisms cause disease, or in many cases it's limited. So proteins haven't been fully identified and characterized that, say, provide protection. So that's hard. Vaccine development in general, I think, takes a concerted effort on what we call antigen discovery. So what are these protein targets, and what makes them good candidates? And it's also very challenging because, again, as I mentioned, you can see great immune responses to these proteins, and you don't see protection. So that part is very tricky. So just because something is giving you an immune response doesn't mean it's going to protect you from disease.
Hi, how are you guys dealing with pH differences in different organs in the mice, and then compared to humans, which is which is the ultimate goal, right?
So I can just talk from our work with animals. What we see is a rapid uptake of these particles by pretty much every cell type we looked at. In vivo, we see a rapid uptake by cells in the spleen, mainly macrophages and dendritic cells. So these are two immune cell types. They're predominant in the spleen. And not only are they taken up by these types of cells, but we know that they essentially end up in a degradation pathway, which is the lysosome, which is acidic. So in In that case, these will just fall apart. For vaccine applications, that's not a bad thing because we want those cells to take them up. But for other applications, that could be problematic. So what we haven't shown you here are efforts that we have ongoing to try to stabilize these particles. So for drug delivery, where you might want to administer these particles intravenously, do we have ways where we can stabilize them so that they circulate longer?
Have you looked into any adverse reactions with pharmaceutical medications that could possibly accelerate the failure of the liver or kidneys? Is that something that you're willing to look into?
So are you asking about liver toxicity?
Yeah, because if there's a significant number of the particles that are continuously concentrated in the kidney and liver, when pharmaceuticals and other medication are being filtered out, it may get clogged up significantly faster.
It's not something we've systematically looked at, but I think that what we do see with all of the animals that we've done, we really don't see any adverse effects at all. We've done both acute toxicity studies, but we've also done long-term studies where we inject the animals and let them go over a long period of time.
I mean, I think one of the advantages is that the nanoparticle is a naturally derived particle. So once you separate it out from its effect, it's recycling in the system anyway. And having too much ApoA protein is actually a good thing for them.
You showed a slide where you coupled the Nano-Bot to a serotonin receptor. Was that receptor functional? 5-HT1A?
Oh, yes, that was yes. So we've done beta-adrenergic serotonin, neurokinin.
So they bind
and
neurotransmitters, produce pharmacology that's similar?
For ligand binding, absolutely. We are working on a paper. So, the receptors sit on the surface. They're actually taken up by the cells. There's internal G-coupled there's other signaling by other proteins in the cell that have to happen to bring about the true pharmacology that you're interested in. And at least for the beta-adrenergic, that seems to be true.
In the cases where there's a good immune response, but not enough actual protection, what's usually at fault there? Is the protein not folded right? Is it?
That's a great question. So we don't really know. It can be different things. So maybe the dose of your protein isn't big enough. Maybe you need a different adjuvant. Maybe you need to administer it a different way. There are a number of things that you can change in that equation, which makes it challenging and fun, but frustrating.
Are there any more questions? Okay, so let's thank our wonderful speakers. I just wanted to let you know that in April, Monday, April 10th, our final talk of the year is on additive manufacturing, and we have Eric Doss, who's a member of technical staff, and Melody Golovic, who's a materials engineer. They're coming to speak, so it's going to be another wonderful talk, and you all have a wonderful evening. I know our speakers will be able to entertain questions, and have a nice evening. Thank you for coming.